Within the cell, there are many different processes that require energy. Some of that energy is a byproduct of cellular metabolism, but there are other ways that the cell has to maintain homeostasis. Free work is performed with no energy input by the cell. Many processes use a combination of free work and metabolic work. Metabolic work is energy input that costs. That means it requires the use of adenosine triphosphate, which is an energy-rich molecule. All of the chemical reactions within an organism contributes to an organism's metabolism. Let's look at some costly work. That is, work that uses adenosine triphosphate. One example is an electron transport chain. Let's unpack this because it can be a little confusing at first. The easiest way to think about an electron transport chain is there's a series of blue pumps. Each one of these blue pumps moves a proton from one side of a membrane to the other. They have different shapes, but they're basically doing the same thing. They're moving that proton, which is represented here by a single hydrogen atom, across the membrane from one side to the other. You can see that that electron, which is here on the far left, travels through and across every one of these pumps and is finally collected at the final stage. As it moves through, each one of these pumps is activated and moves the proton across the membrane. Adenosine triphosphate is a nucleotide. Here you can see the phosphate groups on the far left. Notice how there's 3 phosphate groups. ATP also includes ribose, a sugar, and adenine, which is a nitrogen base. When a phosphate group is removed from ATP, it becomes ADP. In losing that phosphate group, ATP releases a lot of energy. That energy can be captured by living things in order to fuel their metabolism. After losing a phosphate group, ATP is in a lower energy state. It's also helpful to note that there's a single phosphate group floating around. It used to be attached to the ATP, but of course it was removed during ATP hydrolysis. During ATP synthesis, that phosphate group is reattached. Another way to think of this is that ATP is like a battery. When you use the energy within that battery, you lose the phosphate group, and it's like the battery is uncharged. In its uncharged state, it's ADP. To charge ADP up again and make it ATP, you have to add the phosphate group. ATP is used in cellular processes that are costly in work. They cost ATP. Let's look at some free work, things that happen without energy input from the cell. Diffusion is a force that helps fuel many different processes within cells. Diffusion is the net movement of molecules or atoms from a region of high concentration with high chemical potential to a region of low concentration and low chemical potential. This illustration in the center helps demonstrate the flow of energy from an area of high concentration and chemical potential to an area of low concentration and low chemical potential. You can see that the net movement of molecules tends to go downhill on the slope. That is, molecules want to move from an area of high concentration to an area of low concentration. On the far right, you can see a collection of blue dots. They're in high concentration. Imagine that these dots represent molecules of a gas, and the room is the entire slide. What do you think would happen if I released all those molecules? Do you think they would stay on one side of the room? According to this graph, they should move from an area of high concentration to an area of low concentration. Those gas molecules on the far right of this slide are in an area of high concentration. So, if they were released into the environment, they should spread out, eventually settling into a state of lowest chemical potential. We call this diffusion. Here's a thought experiment to help drive the concept home. Let's imagine that on the far left is a cell with a semi-permeable membrane. Inside of it are 2 molecules, those 2 dots. Outside of the cell is a high concentration of molecules. The catch here is that the molecules can't move inside or outside of the cell. but the cell can expand and contract in size. Given that, how can we match the concentration inside the cell to the concentration outside the cell? Do you think that that cell would shrink or expand in order to make the concentration inside match the outside? One way of matching the concentration is to lose water. With the semi-permeable membrane, water molecules can move in and out of the cell, but larger molecules can't. So, in this case, to match the concentration, water has left the cell, and now the cell has roughly the same concentration inside of it as the outside environment. Here's another example. Try to predict the outcome. On the far left is a semi-permeable membrane with a high concentration of molecules. Remember that water can enter or leave the cell. To the right is the concentration of the solution. It's less concentrated than the cell. How would the cell respond to this environment? Do you think water would enter the cell and it would get larger? Or do you think water would leave the cell and it would get smaller? In this case, water would enter the cell, and as the cell expands, the inside environment would eventually match the concentration outside. Tonicity is a measure of the effective osmotic pressure gradient, as defined by the water potential of two solutions separated by a semi-permeable membrane. If a medium is hypotonic relative to the cell will gain water through osmosis. This happens when the concentration of molecules is higher inside of the cell than outside. The effect on the cell is that the cell expands. Another way to remember this is that it expands like a hippo. If a solution is isotonic, there's no net movement of water molecules. That's because the concentration inside the cell matches that outside. If a solution is hypertonic relative to the cell will lose water. That's because the concentration outside the cell is higher. In order to match the concentration inside the cell, water leaves, making the intercellular concentration of molecules higher. Let's try to predict what these three examples represent. On the far left, we see that there's 3 molecules outside and 3 molecules inside. In this case, the solution is isotonic. There's no net movement of water. In the center, we see an example where there's 1 molecule outside of the cell and 3 molecules inside. In order to match the concentration inside the cell, water enters and the cell expands. This would be an example of a solution that is hypotonic. If it helps, you can remember that it sounds like hippotonic. On the far right, we have a solution with a higher concentration outside the cell than inside. In order to make the concentration inside the cell higher, water leaves the cell. That shrinks the cell. We call this hypertonic. Here we can see 2 examples of cells that have concentrations outside that are different than the concentrations inside. When the concentration inside a cell is higher than outside, the chemical driving force goes from inside the cell to outside. If the concentration inside a cell is lower than the outside environment, then the chemical driving force goes inside the cell. Here's a thought experiment. Try to predict the outcome. Remember that osmosis is the spontaneous net movement of a solvent molecule through a semi-permeable membrane into a region of higher solute concentration. It does so in the direction that tends to equalize the solution concentrations on both sides. In the center of the beaker, we have a semi-permeable membrane. That membrane will allow the movement of water, in this case represented by pink liquid, across the membrane. So water can travel to the left side and the right side. The molecules in the concentration, however, can't pass through the membrane. So on the right side, we have a higher concentration than on the left side. If we left this beaker alone, where do you think the chemical driving force would go? Would it go from the less concentrated side to the higher concentrated side? Or vice versa? What do you think this would look like if left alone? In this experiment, water would move to the side with the higher concentration of molecules until the concentration on both sides was even. This is a little counterintuitive because it looks as though the water is defying gravity, but in a way it is. In this case, chemical energy is maintaining the difference in water height on one side of the membrane from the other. Molecules in higher concentration outside of the cell can enter the cell by passing through the cellular membrane through diffusion, or they can pass through special structures through facilitated diffusion. In other processes, molecules are moved against the concentration gradient, but this requires energy in the form of ATP. This is costly work and contributes to a cell's metabolism. These are all key features of cell structure and function, and we'll talk about them more in our next lecture when we explore how cells obtain energy.